आया होगा इस बार। एक बार इसने ना वो बसंत कुल्फी वाला नहीं वो दिखाया था। बाबू यही है, बाबू मान लाइव। तो बाबू मान है।
All the time they are just telling out anything they will service. Like we are waiting, and they are seriously. Like you do surgery in a day, they need to conceive ideas or
मतलब हो गया।
all the time they are thinking like that, walking, talking.
तो क्या करना हमें YouTube पे करना बस। YouTube में सीएमसी डाटा कर तो दिया। कोटा कहाँ नहीं हुआ सर।
उन्हीं जी हो गया।
ओके सर चेक कर लेते हैं।
मैंने सर को भेज दिया, मैं बाकियों को भेज रहा।
ठीक है सर इधर भी भेज दो हम चेक कर लेंगे।
हाँ जी भेज रहा।
ठीक है नया लिंक होगा। पुराना लिंक करके देख, ट्राई करके देख। नहीं डेट होगा ना उसमें।
भेज दिया उन्हीं जी देखो।
हाँ जी सर न्यू लिंक।
Yeah, the YouTube thing is there, it's coming here.
ये ओके पुराना लिंक करके देख, ट्राई करके देख। कह रहा हम लाइव हैं ना। भेज दिया। हाँ जी सर न्यू लिंक।
Yeah, the YouTube thing is there, it's coming here.
पुराना लिंक करके देख। YouTube पे है लाइव।
Yeah, the YouTube thing is there, it's coming here.
YouTube पे है लाइव।
Only we are live on YouTube. Maybe you can mute the speaker when not needed.
Yeah, the YouTube thing is there, it's coming here.
अभी हुआ सर।
Dr. Ashish, we can start, I think. Yeah.
Good morning. Today we have a very interesting topic for the grand rounds, that is genomics of infectious diseases. And we have a very renowned person to speak on this, Dr. Vinod Karia. And before we start the grand rounds and introduction and other things, I request Dr. William Bhatti to just welcome Dr. Vinod and introduce.
Good morning. It's amazing how technology is helpful or influencing medical management. In recent past, we have seen so many changes as far as diagnosis or management of clinical disorder is concerned. And genomics, role of genomics is very important and it's playing a major role. Just talking to Dr. Ashish, the way it is replacing conventional way of diagnosis and management. We are glad today Dr. Vinod Karia is with us. We welcome you, sir, on behalf of Christian Medical College and Ludhiana. And I am sure today's talk will be beneficial for our students and the entire staff. And you will take us into a new field where CNS, staining and other conventional way of diagnosis will be thing of past, and we are entering into a new era. At the same time, I appreciate efforts made by Dr. Jairaj and team for continuously organizing morning grand rounds, which are beneficial for the entire institution. And the way it is improving our learning is amazing. Thank you.
I request Dr. Jairaj to.
Thank you. So it's a great privilege to have Dr. Vinod Karia. I had a brief interaction with them last December, and they had kindly agreed to give a grand round on 2 topics. And earlier we had Dr. Siva Prabhu who talked to us about genomics in medicine. And today it's going to be a very important topic, particularly it is so relevant during this ongoing pandemic. And we are getting so many new variants, and the technology has to be, keep pace with all the changes that are happening in this management and diagnosis of COVID. So I will ask Dr. Ashish to formally introduce Dr. Vinod Skaria. Thank you.
Thank you, Dr. Jairaj. Dr. Vinod Skaria did his MBBS from Government Medical College, Calicut, and then he went on to do PhD. He is a principal scientist at CSIR Institute of Genomics, which is a Government of India institute. He is an adjunct professor at Indraprasth Institute of Information Technology, Delhi. Lot of things to say, many pages. He is a clinician turned computational biologist. His research interests are in understanding the function, organization, and regulation of vertebrate genomes, and how genomic variations could potentially impact them. He is a co-founder of Genomics for Understanding Rare Diseases, India Alliance. His research has contributed to a number of insights and technologies which are widely being used in India for diagnosis and screening of genetic diseases. He has been part of collaborative genomics projects aimed at understanding the Indian and Asian genome diversity. And recently, the COVID-19 pandemic, his lab has seen the genetic epidemiology of this COVID, the SARS-CoV-2 virus in India. His lab has today hosts the major results enabling genetic epidemiology as well as functional interpretation of this coronavirus genomes and the genetic variation. He has 170 research publication in high impact journals. And he has got the Young Scientist Award by the CSIR for his biological science, work on biological sciences in 2012. And he's been elected as a fellow at the Royal Society of Biology in 2019. He's on various editorial boards, and he's got many awards, fellowships, and he has patents also for human microRNA targets in HIV genome and the method of identification thereof. And his publications, you just type his name and format, so many publications would come up. Dr. Vinod, I have a problem. I had a patient yesterday who had taken ATT for almost 9 months, young boy, and he comes back again with a recurrent disease. He has a relapse. He has a fungating node there sitting, and what is the he had taken religiously the medicines, but still he has this disease again. So does genomic has any role towards this identification of the organism again and management? We would like to have your opinion towards the end of your talk. Thank you for accepting our invitation and agreeing to speak to us. Thank you.
Thank you, Dr. Ashish, and thank you, Professor Pandian, for inviting me here. I think my slides are visible.
So visible, they are visible.
Yeah, sure. So over the next 40 odd minutes or so, let me try to convince you a few salient points about how genomics can actually enable the understanding of infectious diseases. I will have a short flashback of around 10 to 15 minutes into what we have done in the past, and mostly restrict my talk to the present COVID pandemic and how genomics has actually impacted understanding of the COVID pandemic globally as well as in India. But before I start my talk, let me first introduce what we work on otherwise. I come from the CSI Institute of Genomics and Integrative Biology, which is one of the constituent labs of the CSIR network, the Council for Scientific and Industrial Research. We call ourselves genome informaticians, which necessarily means that we work on computers on trying to understand genomes. We look at genomes on computers. In the lab, we try to understand a variety of aspects of genomes, which includes how they are regulated and how they are organized. We work quite extensively with a large number of clinical collaborators working in the area of Mendelian genetic diseases. And I'm sure you have heard from Dr. Sridhar Shivasubbu, who is also a co-founder of the Guardian Network. We work quite extensively with country-wide population scale genome programs in Southeast Asia as well as the Middle East in trying to understand the epidemiology of genetic diseases. And a very small part of my lab used to work in the area of pathogen genomics. We used to earlier work in the area of Hologenome sequencing, looking at tuberculosis. But of course, with the current COVID pandemic, we ventured quite extensively into looking at SARS-CoV-2 genomes. So that's what most of my talk will encompass. Of course, you could find more information about what we do on our lab websites at CSR as well as our personal lab websites. Now, for microbiologists out here, I mean, we should all be thankful to people who actually created tools which enabled us to study microbes. And probably one of the pioneers in this area was Antony von Leeuwenhoek. I'm sure medical students and all faculty members would already know this name. He was a scientist, but otherwise a merchant and a municipal official in his day life. And what he created was quite remarkable, was essentially what you see in here, a microscope, which can essentially look at organisms at much greater detail. So most of his work in the 1600s entailed to actually pictorially depicting what he saw on his handheld microscope. But of course, from a handheld microscope, microscopy has evolved over the last 400 or so years. And the first was essentially the microscope as we use today, which was essentially the inventions of Abbe and Zeiss. And then, of course, with higher resolutions, replacing photons with electrons, adding fluorescence, and so on and so forth, gave rise to a variety of other microscopes, including the electron microscope, the confocal microscope, and today what we can do is we can even do live cell imaging. Now, what I'm trying to allude to is, I mean, of course, an irony that practically technology always enabled miniaturization of things from telephones to camera phones to whatever. But this is one field where technology advancement, ironically, has resulted into larger and larger machines. But of course, with the advantage that you can obtain much higher resolutions. But there is a real big problem with this kind of an approach. And one of these major problems is that in at least clinical settings, microbiology is heavily dependent upon a priori information. Or in other words, if you give a sample to a microbiologist and tell him, figure out what is in here, very unlikely that the microbiologist will be able to figure that out because of the fact that there are very limited methodologies for observation, isolation, or for that matter, culture of organisms. So unless you really know what are the spectrum of organisms, you cannot really put on an accurate culture condition to able to isolate or grow them in near pure fractions. And therefore, most of the identification is also phenotypic based, depend upon what culture it grows, what is the culture morphology, and so on and so forth, and much less molecular in our ability to understand what is the pathogen. But fortunately, what we know is that practically all organisms that we have on Earth have one or the other nucleotide material as the genome. So this could be DNA or RNA in at least some viruses. And nevertheless, since these are nucleotides, they could now be sequenced. And then therefore, I surmise that sequencing of an organism could probably be a meta-microscope. And fortunately, we are also in one of the most exciting times in the age of sequencing. As you see here, the cost of sequencing, and mind it, on the y-axis is a log orders. The cost of sequencing has actually been dwindling quite very rapidly, at least in the last decade and a half. And so necessarily, this provides extremely new opportunities to understand pathogens at a scale and resolution which was not possible previously. In other words, I would try to say that if you can marry genomics and microbiology, you can actually have a much better microscope. And this much better microscope is not about visualizing the organism, but actually understanding the genome of the organism. And it's not just about the throughput of sequencing that is going to matter anymore, but also computational tools which can now sift through this enormous amount of data and figure out what is the pathogen in there. And in many cases, not just about what's the pathogen in there, but also be able to assemble complete genomes of the pathogen. And so this is a slide from quite way back. To be exact, this was sometime in 2011 and 12, when Sridhar and myself sort of came across this very interesting paper which described what is called a hologenome. And a hologenome, by definition, as defined by Rosenberg and Rosenberg, is the sum of the genetic information of the host as well as the commensals and the microbiota. Or in other words, all humans are essentially mixtures of human genomes as well as the organisms which reside inside us and also over us. So therefore, we sort of termed this quite quickie term called whole-genome sequencing. In other words, if you can actually take a mixture of organisms and sequence, you should be able to understand what is in there and what are the proportions of each of them in there. So that's how we sort of coined this term whole-genome sequencing. And of course, the key for such an approach was essentially a method which doesn't rely on the ability to ascertain an organism from reference sequences. So typically what we do in whole of genomics is to be able to map it to one of the other reference genomes to understand what are proportions in there. But assume that you don't have a reference genome, you need to have methods which can delineate organisms in a mixture. And this is essentially a synthetic mixture. What you have here is essentially a virus at different concentrations and the E. coli genome, which is a bacterial genome. And what we show here is that you can deconvolute each of these organisms because of very unique features of the genome size, very unique features of the copy number differences between the genomes that you obtain from genome sequencing. Now, of course, one of the applications of this is to identify pathogens in mixtures of samples when you really don't know what is really happening. And we were fortunate to have met with a quite interesting colleague and collaborator, Dr. Shahul Hamid, from the Abdul Hakim College. If many of you have been at Vellore, you would cross this college while going to Vellore. Now, the problem here was quite remarkable, nothing clinical. So India is one of the major exporters of shrimp, and if you have been in Tamil Nadu, you will realize, or Andhra, the coastal belts, you will realize that there are a lot of shrimp farms. And the problem with shrimps in the quite recent past has been that there is a disease called the white spot. As you see here, the shrimps develop white spots all over their body and they die. So the problem here is that there is an organism, a pathogen, which actually kills the shrimps. And this is enormous loss to the industry, not just in India, but also globally. Now, earlier, this white spot was tested using a PCR kit for the white spot syndrome virus, or WSSV. But nevertheless, suddenly this kit stopped working, but nevertheless, the shrimp was dying because of white spot. So we jumped in and sort of applied this technique over here. And the technique in essence is very simple, that you can take an infected shrimp, you can grind them up, and of course do DNA isolation, sequence the DNA, and now put it through the computational pipeline, including reference mapping, as well as the method that I sort of mentioned, to understand what is in there in the pathogen. So this is the map of the reads, and what you see here is a quite distinct peak at a coverage of around 500 and 600. And you could take these reads, and you know that this is looking like a virus because it's a quite small peak. The larger peak that you see here towards the lower coverage is essentially the shrimp. And what you could do is you could take this reads and sort of assemble into a almost near complete genome of the virus. And what we established is that this is indeed white spot syndrome virus, but different from the previously known genomes of white spot syndrome by a few nucleotides. And in fact, we could also map that there were a number of deletions which map to the white spot syndrome virus. Now, incidentally, what we also understood is that the diagnostic primers for RT-PCR, though they were built on quite consensus or conserved regions across the genome, mapped to one of such deletions, and therefore they stopped working. In other words, now this is a fantastic opportunity because now you have a technology and a tool which can now identify a virus even without knowing the reference sequence of the virus. And this was done in collaboration with Dr. Shahul Hamid and was published late back in 2018. Of course, there's a lot of work that has happened in between, including going back to the archival samples for almost a decade and trying to identify at what point in time did these deletions occur, where were these deletions imported from, how did it spread across the different parts of the country, etc., etc. But nevertheless, now we have a technique which can identify the deletion, and of course, you could use this information to build new diagnostic primers across the genome where there are no deletions. But let us now fast forward into 2020, and I'm sure this is a period in time when genomics has become the mainstay. Even newspapers have been writing about genomics and genomic variations right there in the front page, and thanks to all the efforts by Chinese researchers who put the first or the initial genomes of this virus in public domain early in January 2020. Now, practically all that we know and all of genomics on SARS-CoV-2 is thanks to this genome, which we call as the WH-human1, which was isolated from a patient from the seafood market in Wuhan. Now, while this is published in Nature, what you also realize by quickly going back to the materials and methods section is that the sequencing of mixtures of genomes have actually become the mainstay. And you would see here that at no point in time was an attempt even made to isolate the virus or culture the virus or visualize the virus in electron microscopy, which all formed the mainstays of conventional virology or conventional microbiology. And what really happened in this particular case was that the sample, the bronchoalveolar lavage, was directly processed for iron isolation and sequenced. And what followed was an extensive array of computational tools to not just assemble the genome, but also put them in the phylogenetic context of other coronavirus genomes, which has been sequenced and reported from elsewhere in the world. Now, you would all ask this question, what is so unique about coronavirus, or for that matter, any virus or any organism? And all organisms evolve by virtue of mutations, and this includes humans. We also do evolve. Compared to viruses, we evolve at a much, much lower rate. And even between viruses, there are different viruses which evolve at different rates. And for coronavirus, it's estimated that approximately 1 in every 7 to 10 days, there is a new mutation that occurs in the virus. And this has a lot of implications. One, implications in the evolution of the virus, in its ability to spread faster, wider, and also evade many of the immune responses. And it has also implications in our ability to trace the spread of the virus, or what we call as genetic epidemiology. I will come to that in a few slides from now. And this is sort of exemplified in here. So what I've put as an example are just 3 genomes from very early in the pandemic, all from China, right from the first genome that I sort of mentioned, another genome which was collected from Jan 8th, and a third genome which was collected from Feb 27th. And what you see here is that even within this very short period of time, the virus has accumulated mutations, and accumulated mutations on a genetic background which already existed. Like, for example, the second genome has 1 genetic mutation, the third genome has the genetic mutation, and additional set of mutations which have occurred in the genome. Now, this is very important because this allows you to understand and trace the spread of the virus or the lineage of the virus as it spreads across different parts of the world. And the second big paradigm that has happened in this particular pandemic is that this data is widely shared and widely accessible. And never in the history of mankind, we had almost now close to 600,000 genomes of an organism sequenced and made available by researchers across the world. And most of these genomes are today available accessible for free. You can download and analyze them, and they're available in one of these public databases, whether it is GISAID or the NIH Genbank, where you can download and access not just the genome, but also a lot of metadata which can enable deep analysis of these genomes. Now, what you need to keep in mind is that virus evolves by mutations, and mutations occur, or mutation substitutions occur at a very constant rate. I sort of mentioned what this rate is all about. Now, as the virus mutates and gets transmitted from individual to individual, that's how they spread across the population, they accumulate mutations on the genetic background. So for example, if the virus has accumulated one mutation, that is for convenience, mark it 1. Over the next 7 days, it would accumulate another mutation at another different position, that is for that matter, mark it 1, 2. And if that 1, 2 got transmitted to 2 individuals, the third mutation could occur at the 4th position, so that would become 1 to 4, and or at the 3rd position, which makes it 1 to 3. And at this point of divergence, of course, they could accumulate further mutations as they move forward. Now, if the mutation rate is constant and the spread is also a constant, then across time, if you sample the virus in a particular population, you would expect to have equal proportions of all these mutations to happen, right? I mean, that's how it should be. Or for that matter, if you take a time point, you should have 1 to 3 and 1 to 4 as the same, because they have diverged some point of time. They have a constant rate of mutation and a constant rate of spread, then therefore they should have equal numbers. But that's not really what happens in real life situations. At some point of times, you will have 1 proportions of lineages. For that matter, it is mentioned 1 to 4 was found at a very large number compared to 1 to 4, 1 to 3, or 1 to 3, 5. Now, what does this matter, and how does this matter? So these are events, what we call as emergence events. Emergence events could be because of multiple different factors. The human factors would be the most important, because this would be because of super spreader events, where an individual would transmit the virus to a large number of individuals, and then therefore a disproportionate large number of individuals with a particular virus. The second possibility is that you could have a viral genetic mutation which could enable faster, wider spread of the virus, and then therefore it can spread much more effectively compared to the other viruses out there. So these are the 2 factors that you need to keep in mind, the human factors as well as the viral genomic factors, which can explain such kinds of emergence. Of course, there are many other methodologies to delineate whether it's a human factor or a mutation. The other thing that you need to keep in mind is that when we talk about SARS-CoV-2, these are all genetic variants which are different from each other by just a handful of genetic variants. To be exact, less than a dozen in the recent times. They're not really different strains of the virus. So when we call a strain, we typically call SARS-CoV-2 as a strain which is distinctly different from the Middle Eastern Respiratory Syndrome or the MERS-CoV, or the initial SARS-CoV, which all caused outbreaks locally as well as regionally. So mentioning these lineages as strains or a new strain of the virus is probably a wrong terminology to use. Now, of course, with this global data, there are also phylogenetic maps of these genetic lineages as they have spread across the world, contributed by researchers. And broadly speaking, in the older classification, there are around 10 genetic lineages of the virus in the world. And early in the pandemic, when I say early in the pandemic, mostly in January and February, these lineages roughly corresponded to the geographical areas where they had spread out of China, of course. And as you see here, they are all delimited or delineated by different colors. What you see here is a very large cluster of blue dots. So this is what we call as the A2A clade of the virus, which is marked by a genetic mutation, which is D614G. And of course, there is also a lot of literature on D614G and how it causes increased infectivity of the virus and so on and so forth. And of course, there are other colors out here, which is the B clade, which is all in yellow, which includes B, B1, B2, and B4. These are all the lineages which emanated out of China and spread into China as well as into Southeast Asian countries. And of course, there are many smaller lineages of the virus which are in green, for example. This is the A3 lineage, which are the virus which spread early in Iran during early pandemic. So roughly speaking, early in the pandemic, these genetic lineages sort of delineated the geographical regions where they spread, and then therefore, when they were introduced into a new population, they could also serve as a tool to understand where did this individual come from, or where did this virus come from. And of course, in India, the first case of the virus was reported in the southern state of Kerala early in Jan 2020. And of course, over time, the number of cases have increased or peaked across the country. It had indeed come down, and in the very recent weeks, we also see a resurgence of the number of cases in different parts of the country. Now, in terms of genomes, today it's estimated that there are close to around 10,000 genomes of SARS-CoV-2 sequenced in the country, out of which around 7,000 odd sequences are available in public domain. And we have this quite interesting resource, what we call as INDICOV, where you could search for a particular genetic variant or a genetic mutation, and you should be able to understand where in the country do you find such genetic variants. And all this data is contributed by a very large number of researchers and research institutions from multiple different states who have all contributed this data in public domain so that there could be very meaningful assessments of genomic information that could be made. For interested individuals, you could follow this website, which is updated almost on a weekly basis, where you can access the genetic lineages or the variants of the virus and the frequencies. Now, one of the ways we have been able to accelerate the genome sequencing to this large number has been because there have been tools that have been developed very specifically to sequence genomes at higher throughput at a quite good turnaround time and at a cheaper cost than that was previously available. And this was also efforts of Sridhar, whom you have heard in the last grand rounds, who put together this program called the COVID-19 MegaLab. And the idea of the COVID-19 MegaLab was to enable high throughput sequencing of SARS-CoV-2 to enable detection as well as for genetic epidemiology. And now this uses COVID-Seq as an approach, and COVID-Seq is today an FDA approved test for SARS-CoV-2 detection. And one of the first labs even globally to undertake this COVID-Seq approach for genetic epidemiology was CSIR-IGIB. And this was possible with a large number of young students who dared the pandemic and put together this entire protocol, which is quite complicated in many ways. So this can sequence anything from 96 to 3,000 samples in one single go, and each run is approximately around 12 to 18 hours. Sequence the virus and also be able to automate the analysis procedure, which includes detection of the virus and also classification of the virus, including what are the genetic mutations that arose from these samples. Now, this is one approach which really changed our capability to sequence genomes at an unprecedented pace than that previously available. And that also allowed us to collaborate with an extensive large number of states, mostly in the southern part of the country, which includes Tamil Nadu, Karnataka, Andhra Pradesh, Kerala, Orissa, UP, as well as Rajasthan. And what we did was to essentially sequence a large number of genomes from these states at different time points and try to understand how did the virus spread and evolve as it spread across different parts of the country. And one of the remarkable things that we sort of identify from the genomic data is that of the 10 odd lineages that are present worldwide, which I sort of mentioned before, there are around 6 of these genetic lineages which are now found in India, A2A being the predominant one which arose out of Europe. But nevertheless, we also found a very distinct lineage marked here in purple, which didn't really map to any one of the global lineages. Now we sort of named this as A3I, I to signify that these are predominantly found in India, and A3I to signify that some shared origins with the A3 lineage of the virus. And I mentioned before that the A3 lineage of the virus are found out of Iran. Now, we ventured very deep into understanding this particular genetic lineage, and this genetic lineage was marked by 4 genetic mutations, and of course, it had a mutation not absolute numbers, but also shared in some way with the A3 lineage of the virus. So this is what you need to also keep in mind, that each of the genetic lineages are marked by very few number of genetic mutations. So therefore, you should not call them as strains of the virus, but only as clades or mutants or lineages of the virus. Now, what you can additionally glean from the information available is the mutation rate of each of these clades. And what we found is that the clade IA3I has a much lower mutation rate compared to the prevalent clade, which is the A2A or the other genetic lineages out here. Now, this has 2 implications. You could imagine that if a virus has a much lower frequency for substitutions or frequency for mutations, then it has a fitness disadvantage, and therefore we could expect that a virus with a much more mutation rate could now overcome the pandemic in a particular region. And that's what we find here. So the bottom right panel essentially suggests that the virus, while it emerged early in the pandemic during March, eventually faded out, and we don't find that virus lineage anymore in the country. And that has largely been superseded by this blue space, which is the A2A lineage, which today is the predominant lineage of the virus in the country. And of course, you could make a lot of analysis in terms of divergence estimates, and what we suggest is that this virus seems to have diverged from one single central point across to different parts of the country. In other words, what we suggest is that this is a super spreader event which caused the virus to spread to a very large number of individuals who took it to across the different parts of the country. And you could also, from the epidemiology data, look at the proportions of these clades in different parts of the country. And what you see here is that the virus is predominantly found in Delhi, and of course some southern states like Telangana and Andhra Pradesh and Karnataka, but found almost 0 in some states like, for example, Kerala, where you didn't find any single case with this particular genetic lineage of the virus. The other thing that you can do with such genomic data is that you can also sort of draw contact maps or molecular contact maps. And this is very important because contact mapping is a process in epidemiology where you can essentially interview an individual and understand who are their contacts. But as the pandemic grows in number, and of course people travel from a place to another place, you practically have lost the ability to do contact tracing or contact mapping. And that's where genomics can become helpful, because looking at the genetic mutation of a particular virus, you can sort of trace that virus from a point in origin where there is some kind of a prevalence. Or in other words, from district to district, from state to state, you can sort of plot the map of the virus transmissions. And this is what you see here. You could see the virus moving from east to west, rather west to east, and from north to south, and as it progressed across different phases of the pandemic. In other words, what we also suggest is that there's a significant spread of the virus in India, which has been contributed by interstate travelers. And this is what you also see here in a very different format. Now, each of these clusters or the blobs out here signify the number of genomes that have been isolated, and they're very close together in terms of similarity in the genomes. And nevertheless, these are all local clusters of or super spreading events that has occurred in different parts of the country. Or in other words, what we suggest is that a significant large amount of the local spread of the virus has happened by local clusters of super spreading events, which, like for example, if you see here in this particular picture on the right side, you have this very big cluster, and this big cluster has contributions from Telangana, from Tamil Nadu, and so on and so forth, and has diverged out into many different states like Karnataka and Kerala. And this is essentially how the local super spreading events occur. They occur in a point in place in time, and then spread across with travelers who travel out from that particular location into different regions of the country. So in many ways, you can actually map the virus as it spreads across the country, and also map not just the origins of the virus, but also in many ways be able to trace how did the virus get transmitted across a particular region. For example, if you see one of these clusters in this particular case, green in Telangana, this is all one single cluster of the virus, all from one single place, but would have originated from a central cluster of people who were super spreading at this point in time. Now, this is all about epidemiology, but the viral genome sequence can reveal a lot of more information which can be very useful to build better diagnostics, or in many ways evaluate diagnostics, to understand immune epitopes. And this is very useful because we need to build better vaccines, and also understand very pertinent aspects about the functional effects of these variations in the spread of the virus. I'll touch upon one of these aspects. This is about evaluating and building better diagnostics. And as you know, that real-time PCR is the gold standard today for detection of virus across the globe. And real-time PCR depends upon the principle of PCR, or polymerase chain reaction, which necessarily means that you have DNA probes which can identify a very specific region in the viral genome, amplify that region, which can now be probed using another set of DNA piece which has a fluorescent label. Now, the efficacy of PCR depends upon the ability to specifically bind to the viral genomic material or the amplified product of the viral genomic material. Therefore, genetic mutations in these primer binding sites or probe binding sites could affect adversely the efficacy for detection using a particular kit. Now, to understand this, you need to have two things. One, the primers and the probes and the sequences, which in most ways is an easy task because they're available in the kits or the inserts in the kit. And the second thing is genetic variations, which also fortunately is widely available through databases, which I sort of mentioned before. So what we did was quite simple, and this was done by a set of undergraduate students. They collected primers and probes and the sequences of primers and probes from publications, from websites of diagnostic kit manufacturers, from package inserts of diagnostic kits, in many ways from public resources, some of which have compiled such primers and probe information. And we map them back to the viral genome and asked a very simple question: are there genetic variations in this primer and probe binding sites which are frequent in the population and can change the efficacy of binding? And you can calculate the efficacy of binding by thermodynamic parameters, what we call the Gibbs free energy. And the panel that you see here in the bottom, the red dots are essentially the variants which map to the primer sites. The y-axis is essentially the frequency of these variations in the general population. So what you see here is there are some genetic mutations which are very frequent in the population and can also disrupt the primer binding efficacy for a particular kit. This is not for all kits, for a particular kit, because typically a kit would have 3 or 4 sets of primers specifically targeting viral genomic material. So in other words, even without doing a test, you should in many ways be able to ascertain whether a kit is likely to be working in your population or not likely to be working in a particular population, given the information about the variant frequencies in your population. And of course, the other aspect that you can sort of use genomics is to address very pertinent questions in immunology, and of course, what we call as clinical conundrums. And there are 3 clinical conundrums that you come across. One is of persistent infection, the other one was of reinfection, and the third one is of reactivation. Now, to understand this, you need to also briefly understand the immune system. And briefly, the immune mechanisms against SARS-CoV-2 are 2-pronged. One is a humoral immune system, which is modulated by B cells, which produce antibodies. And of course, you have a cell-mediated immune response, which is modulated by T cells, which cause cytotoxicity. Now, these 2 mechanisms are essentially what prevents infection and reinfection and control of an infection in a particular individual. Now, clinically, these 3 states sort of occur, but they're indistinguishable from each other just by clinical examination. So you need to have a better approach to understanding this phenomenon. And before I delve into that, one of the opportunities that we had early in the pandemic was to set up this kind of a surveillance system in a medical college. And the idea of creating this surveillance system was to put every single healthcare worker who was on COVID duty on a regular surveillance of around 1 in every 15 days, where an RT-PCR sample would be taken from these individuals on a constant basis and be tested. Now, this surveillance brought us quite interesting insights, and one of these insights was 2 individuals who tested RT-PCR positive during the surveillance mechanism. And of course, during the protocol, that period of time, these individuals were hospitalized because that was a protocol early in the pandemic, and they were discharged when they became RT-PCR negative. But at least 2 of these individuals became RT-PCR positive at a later point of time, and during the time, depending upon the protocol, they were either hospitalized or they were kept in home quarantine. So what we need to understand is this a case of reinfection? Is this a case of reactivation, or is it a case of persistent infection of the virus? Now, of course, RT-PCR negative reports could suggest that this is not probably persistent infections, but as you would know, RT-PCR results, a negative result is not really suggestive that there is no infection. It could then therefore be either reactivation or reinfection. And the only way to sort of prove this point was to take the samples of the PCR positive samples at both episodes. So this is the genome sequence of episode 1 and episode 2 for the 2 individuals I1 and I2, and map the variants for these 2 genomes and ask this question: are they the same virus or are they different viruses? And of course, as you can see from here, both the episodes E1 and E2 in both the individuals had distinctly different genetic mutations, which sort of suggest that this is a case of reinfection. Now, this was a case of reinfection, and these individuals were asymptomatic, and they could be picked up only by a surveillance mechanism. And then therefore, we also had the opportunity to suggest that asymptomatic reinfections could occur, and these individuals would otherwise never present to a clinician, because without a clinical feature, they would never go and get tested. So therefore, this is also globally one of the first reports of asymptomatic reinfection in an individual. And what we also found is a mutation, N440K, in the spike protein. I will come to that in a few slides from now again. And this was another patient. This patient was referred from Medanda. And the idea of referring this patient was that this patient was on chemotherapy. And as a routine protocol before chemotherapy, these patients were tested. And this patient was tested at some point of time as RT-PCR positive. The chemotherapy was deferred and was restarted again when the patient was RT-PCR negative. But after around 42 days, the child tested positive again just before chemotherapy. And then therefore, this was perplexing, because the question is, why did this occur? So the 2 episodes, E1 and E2, were sequenced, and as you see here, both E1 and E2 has the exact same complement of genetic mutations. So in other words, this is the same virus which seems to have persisted in the child all throughout during the infection. So this also therefore becomes the first case of a reactivation of the virus, because the virus is essentially the same. The virus would have gone underground during the phase of chemotherapy, but nevertheless got reactivated at some point of time. So this also becomes the first case of genomically characterized reactivation of the virus during SARS-CoV-2 pandemic. Now, the other aspect that you need to keep in mind is one of the functional sets of variants that the virus is accumulating, at least in the recent months, is a class of variations what we call as antibody escape variations, or it's otherwise colloquially also termed as immune escape variants. Now, as I said before, humoral response is one of the arms of the immune response, which is important for containing the spread of the virus, or for that matter, the early infection of the virus. And now it is estimated that there are around 140 odd variations, which has the property of antibody escape. And this could be against very specific monoclonal antibodies. This could be against polyclonal pool of antibodies. This could be against convalescent sera, so and so forth. Now, depending upon the effect of these genetic variations, you could also classify these genetic variations depending upon how severe could be the immune escape phenomenon. Now, individually, these genetic variations may not be of very great concern because they could disrupt one or multiple of the monoclonal antibody sets, or in some cases, at least for one of these variations, polyclonal response. But what you really need to be worried about is the combination of such variations co-occurring in a virus, which could now obviate the action of a very wide variety of antibodies, and then therefore they could escape the polyclonal response. Now, incidentally, the virus also has an Achilles heel, and the immune escape is essentially that Achilles heel, because of the fact that almost all of these immune escape mutations occur on the receptor binding domain of the virus. And the receptor binding domain on the spike protein is here marked in blue. And practically all these mutations do occur in the receptor binding domain. So in other words, the virus really cannot mutate much more further without losing its ability to latch onto the human receptors. In other words, the virus has to strike a balance between its efficacy to latch onto the human receptors and also escape the immune phenomenon or escape antibodies. Now, depending upon how many antibodies do they escape, there are very specific locations in the receptor binding domain that it can mutate, and therefore we also believe that this number is a finite measure. The virus cannot really mutate at all positions to escape the immune system. There are only very small and finite positions in the genome that it can mutate to escape antibodies. So in other words, this is a very useful information to build better vaccines which can avoid this phenomenon. Now, if you ask this question, are these immune escape mutations prevalent in the population? Not very prevalent, but there are at least 19 mutations which are now found in India. And one of these mutations is what we call the N440K, which is found in a very large number in the southern state of Andhra Pradesh. And of course, now this has been reported in many other southern states, which including Karnataka, Telangana, and of course, now very recently in Kerala. The other major immune escape mutation, which is not there in this particular table, but recently emerged in the last couple of months, is another mutation called the E484K. Now, E484K can escape multiple antibodies, can escape convalescent plasma, and can also escape the vaccine sera. So we have recently found this mutation, at least in public, a few public genomes which are from Maharashtra. We don't really know the breadth and spread of the virus yet, which is a work which is continuing. Now, can immune escape mutations cause reinfection? The answer is yes. There is at least anecdotal evidence which suggests so. And what we also suggest in a case of reinfection, and this is from Kurnool in Andhra Pradesh, a collaboration with Kurnool Medical College, an individual, a male individual who tested RT-PCR positive at 2 episodes. In the second episode, he was symptomatic. The viral genome sequence suggests that he had distinct mutations, 14 mutations, which are shared between the 2 episodes, and N440 was one of them. In other words, this is the second case of a reinfection with N440K genetic mutation. Now, there are around 50 odd genomes of reinfections available globally today, and there's been no other mutation which has been recurrent and has caused a recurrence of reinfections other than N440, which belongs to the immune escape category. Now, you might ask this question, probably COVID is over and the vaccines are in place. At least we are almost close to reaching 1% of the population to get a vaccine. Now, is genomics any more relevant? The answer is probably yes, because of 2 reasons. One, despite a high level of vaccination, There have been cases of breakthrough infections that are happening. So we need to track and prevent and understand such breakthrough infections from emerging, because one of the concerns that we would all have is that they would have genetic mutations which can now evade the antibody response. And therefore, any emergent infections of individuals who have completed the course of vaccines, and today the healthcare workers have all completed their 2 courses of vaccines and have also completed 2 weeks after the second course of the vaccine. So any emergent infections in these group of individuals, we need to closely watch. We need to probably sequence all of those viruses to understand what are the genetic mutations which enable the virus to escape probably a vaccine response. And this is very important because we need to understand it fast before it can really spread into the population and can make vaccines ineffective. I think I'll stop there and take questions, but before that, let me acknowledge a lot of people who made this possible. The NGS and the COVIDSec and the MegaLab was all put together by Sridhar. You heard him in the last talk, and a fantastic set of young researchers in the lab. A fantastic set of collaborators from across the country, from multiple different states, who put together the samples for genome sequencing during the pandemic. And of course, a wonderful set of researchers in the lab who put together the annotation resources, the databases, have been collecting data consistently all over the pandemic. And of course, this is all possible with data contributed from a large number of researchers across the country and across the world, and of course funding, consistent funding from CSR, which made this all possible. I think I'll stop there and take questions, but of course there's some states at least today which has taken this forward in a very great way, and one of these examples is Kerala, which is a consistent surveillance program using genome sequencing, covering all 14 districts, 100 samples which are being sequenced every month. That's roughly 25 samples every week. And to use this data to not just validate policy, but also to build better policies and to understand emerging lineages of the virus. I think I'll stop there, take questions. Thank you.
Thank you, Dr. Vinod, for that talk. And we have few questions. Before I go to the questions in the chat, does anybody have any questions here? Especially the medical students. Yeah. We can get this mic there, please. Shaukat, mic. So we have a question from the microbiologist.
Thank you, sir. It's more a thank you to IGI. I'm part of the molecular microbiology team, and 2 departments have come together, Medical Research Innovative Center and Molecular Microbiology. And we have just 10 months experience with COVID. And recently, Dr. Agarwal, who's the health advisor to Punjab Health Department, and Dr. Pastor in ICMR, had called all the representatives of the labs, and they've said that all the mild diseases with PCR positive and ICU or death, we have to separate out, and we have started doing it from October, hoping that northern India also the mapping will be done, sequencing after sending it to IGIB. So we are awaiting the official letter. So our lab has already started doing it. Thank you for the initiative from IGIB. And one question is, sir, with your experience, is the mutations more in the spike protein so far in India?
No, the mutations are not in the spike protein. Most of the mutations are not in the spike protein. Mutations are spread across the genome, and that is natural because the virus keeps mutating at positions where it can mutate, and this is essentially a random process. Now, having said that, the spike protein mutations are important to look at because of 2 reasons. One, that is the exposed protein of the virus and therefore the target for the immune response. And secondly, that is also the exposed protein of the virus which latches onto the human receptors. Now, because of these 2 reasons, spike protein mutations are of great importance. And for that matter, 70% of the functional mutations on SARS-CoV-2, which has been experimentally validated, belong to the spike protein. Now, doesn't mean that other mutations have no effect. Of course, there are other mutations which do have effect, which includes mutations in a gene called ORF8. And there have been consistent evidence across, not just for this coronavirus, but also other coronaviruses, the deletion mutations in ORF8 could actually attenuate the virus. And we do find these mutations globally as well as in India.
Thank you. Because our RT-PCR kit actually looks at E gene and the RDRP and the N gene. We are not looking at the S gene. So
right. So the N gene has a lot of mutations, and what we also show is that there are a lot of mutations which can actually disrupt the primer binding on the E and the N genes.
Thank you, sir.
There are plenty of questions here. Anybody else has any questions? Okay. So, let's say, can you enlist various mutations responsible for lethal mutations in COVID-19?
Okay, so I mean, almost everybody is interested in understanding, is there a mutation of the virus which can actually make it more lethal, right? Now, frankly speaking, there is no mutation yet which makes a virus more lethal. But having said that, you need to keep another concept in mind, that assume a virus has X percentage of lethality, right? For that matter, let us say that it is 1%. Now, what does that mean? That means that 1% of infected individuals would have mortality. Now, 1% of infected individuals, in the term of the denominator, the infected individual number would change quite drastically for a virus which can become more infective, right? Or for that matter, if you ask this question, why are we really concerned about the B1.1.7 or the UK variant? It's not because it is more lethal. It is just because it can spread much faster. And if it can spread much faster, then necessarily means that the number of individuals who could be infected becomes large, right? And the number of infections becoming large would also mean that 1% population in absolute numbers become large, right? And then therefore, the virus in essence in clinical observations would cause more lethality. And that's what we are really concerned about. So to answer that question, there is no viral mutation which can increase the clinical symptoms of the virus, but there are mutations which can increase or decrease the infectivity of the virus, and this can significantly influence the absolute numbers of mortality.
Okay, one more question. How does the virus go underground and escape detection by PCR, referring to your reactivation slide?
Yeah, so we don't really understand that yet. And frankly speaking, we don't have the tools to understand that in that great detail. What we can only surmise or hypothesize is that the immune system could have gotten hold of the virus and sort of contained the virus. But of course, this kid was on chemotherapy. So during the chemotherapy, probably, which is also including immunosuppression, the virus containment was not really effective at that point of time. That's all what we can hypothesize. At this moment, we still do not have any clue. But having said that, we have now investigating one more case of a similar reactivation, but this time in an individual who was apparently normal, I mean, not on any medication. So frankly speaking, we don't really understand.
Okay. Is reactivation seen in healthy individuals?
Yeah, so the quick answer is yes, we have now an individual with a reactivation who is otherwise apparently healthy, not on any medication, not with any comorbidities.
I think we can take this as last question because there are plenty of other questions and time is up. Is the decrease in dengue virus cases related to COVID-19 pandemic?
Oh, very interesting question. So we have been essentially tracking the reportable outbreaks over the last 10 years, almost a decade. IDSP provides this data in public domain. And what we found in the last 1 year is that practically every other communicable diseases have actually taken a significant dip. And the dip is almost 80%. Now, there could be two reasons for this. The first reason is that there is indeed a dip because people have been keeping indoors, schools are not open, the transmission of the infection is meager, so on and so forth. The second possibility is that just because everybody is busy in COVID, nobody is even looking at them or even reporting them. So we are yet to understand what really went wrong, but yes, indeed, in numbers, absolute numbers, there has been a very significant dip in 2020 compared to the last one decade.
Thank you, Dr. Vinod, for taking your time for us and giving this excellent presentation and taking all the questions and answers. And hope we can hear from you again sometimes soon with more of bacterial genomics. This time it was mainly focusing the viral genomics, the bacterial. Will the culture plates still exist, or they will be obsolete? Or how will the samples be reaching the lab? 24 to 48 hours, is that the waiting time? Or we can get the reports quick by your genomic method? Or how do you prevent overgrowth and so many other things, which I had thought of asking and hearing from you. Maybe a next lecture sometime in future. Sure. Thank you very much.
So if you have pending questions, you could also mail me directly, because I understand there are a lot of more questions. So you could probably mail me directly. I will be more than happy to answer them.
Yeah, next time we can take, because we all have our clinical responsibilities now. We need to carry on. Thank you.
Thank you.
Thank you very much.